stuff.
Some debris.
Deep
could
sea,
be the dead base of a
stuff. Yeah.
or something.
Go ahead.
It's an interesting round circle, top right.
Ship move.
Copy that,
20 meters,
I think
bearing
it's a sponge
210
base.
degrees. That doesn't.
The one we just saw, yeah.
Yeah, I think that.
Oh, and they got another
rock
coral
feature we're
coming up
looking at
here
is
on
the
the
one.
right.
Too.
One
Yeah,
of the
it's
Treatoplura
nice.
glass sponges to the left, but let's go for the corals. Got several colonies there, but another one of these, maybe the same thing we just imaged. Maybe oh, there's a colony right in front of that coral
that,
pilot.
thank you.
That's
Yeah.
lyrate.
Looks like a chandelier, like a
Candelabra or
candle.
something. Yeah.
Get
This
right
is
on
going to
top
be
of
yet
it.
another different species of octocoral.
Also a different black coral in there. Got some
Oh, yeah.
sparse branches.
All right, and look at the way the animals are picking up now they're on this consolidated substrate, Scott. Good sight. Nice pick here. We're off to a great start.
Okay, Copa, can you check my
Thanks. And
rotator?
also, this looks like it's a
Video
local
can't
high
get
point.
super close
so
quite
this
yet,
consolidated substrate
but
is
I
also
will
raised, which is
couple
another
minutes.
thing that
So you
accelerates
can
the
come
current,
down,
and
go
where you'd
for
expect
that
to see the largest
candlestick
of the
guy.
central fauna.
Do you want to start?
Unless,
Yeah,
of course,
yeah.
the currents get too
You can,
strong, they can't get too big,
Okay,
because then
just
the
got my
colonies
foot
might
down so you
break.
can rotate down a little bit here.
Coming down.
Yeah, video, you can come in. I think this is what they wanted to look at first.
That definitely
I
is.
also saw a spiral
So
off
Is
to the
it
right.
centered? Is that where we're?
Yeah.
It's top.
Rotate
There it
up.
is.
Yeah. Yeah, go ahead and come in.
Candelabra looking thing.
Yeah, rotate up and come in. Yeah, there you go.
Is it still up?
Yeah,
Yes.
keep rotating up.
Oh, we're going for that one.
Large
Copy.
colony,
Yeah,
yeah.
I was going for a different
Oh,
one.
sorry.
That's all right.
There you
My
go.
mistake.
Pretty far away here, but once I get more tether, I can get closer.
Okay, so this is going to be yet another species of octocoral. It's in the same family as one of the ones we saw earlier, the primnoid family, but this is different, and my best guess on this would be, what would my, Calyptrophora angularis.
Calyptrophora, you're going to say?
Yeah, Calyptrophora and maybe angularis, Scott? What do you think?
Yeah, well, I'm not as willing as you to go to species because, I see so many different corals that have very similar morphology, and so until we can actually see those microscopic details, I feel a little uncomfortable making that kind of a guess. I also see there's a crinoid there on the left that has maybe a eulimid gastropod sucking its juice.
Oh, interesting.
I'll turn that over
Is
to
that max
Chris.
zoom rolling?
Oh, there it is.
Okay.
Yeah.
Yeah.
took a
Are you
look.
max zoom rolling?
Yes, it's max.
Okay. You can center up that crinoid, John. Rotate up just a smidge.
You wanna-
Stopped.
Chris is logging that already, and there's a brittle star. Or is that
So
not a
that's-
crinoid?
That's perfect.
Yeah, well,
Okay.
the brittle star is up there, but
Yeah.
he's talking about the. So there's a tiny, as Scott pointed out, there's a what's called a eulimid snail, a snail in the family Eulimidae, feeding on the arm of this feather star. What's interesting about this is that this feather star is extremely small. I don't know necessarily if this is a juvenile. I assume it's a juvenile, but it could easily be an adult of a separate, very tiny species. But the snail is attached to it. It's something that we see not uncommonly, but interesting for the size and for the dynamic. Thank you.
Yeah, we're good
Yeah,
for
Chris,
science.
that's interesting. I don't believe I've ever seen a
different,
snail that large in comparison to the
Is the
crinoid
bottom
that it's
base, go
feeding
to the
on.
base.
Yeah, it is a very disproportionate size.
Sorry, do
we
Rotate
rotate
down.
down?
Yeah.
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 Crinoid until that
Video, which one are
crinoid
you
grows
talking
to
about?
adulthood,
Is
and
it
snails
that right there?
stick around
Yeah.
and
Small black
continue to
coral.
live with it.
Yeah,
Very small.
it makes me wonder. There are sea stars that have
Oh, here, it's
snails
just
living
video.
on them all throughout their life.
Yeah.
All right,
Yeah,
pilot,
so
I
this
think I'm
is
starting to
another one of
swing.
these
So do a
heteropathies, I
little
think,
bit
which
more
we've got a
tether.
couple of images of. The different black
video.
coral was off to the left.
I think this is a parantipathes.
Oh, parantipathes? All right, I'm very
Yeah, I'll
sorry.
give you a rotator
There's
back.
a different black coral.
You have it.
All of those pinnules are pretty
All
even
right.
in length. You
Thanks,
don't see
Copilot.
the really short pinnules coming off of one side. And I suspect that's what that sparsely branching thing is going to be in the
Some
back
weird
left as
coral
well.
bottom right also.
Leave it, Charlie.
There's also a weird looking one just behind that dead sponge in front of us.
Yeah, that's the one we're looking at, the kind of leggy thing between the dead sponge and the bamboo coral. I see. Okay.
Kind of thinner.
Keep going a little bit to the right. Yeah, the thinny, wispy thing.
Yeah. Centering up now.
Oh, this is a weird one, Scott. Oh, I think I might have an idea.
branching thing in the
Maybe-
focus?
Yeah, could
Frame it up.
this be Belopathy, Scott? A weird. Well, I don't know. What is this?
Still zooming in.
I've never seen anything like this. I don't know if you have, Scott.
Yeah, I don't know. This might be in the Antipathidae. I suppose it could be. I guess it's an Umbellapathes. Maybe we're just looking at it at a funny
Slight
angle.
tilt up.
I
Yeah.
don't know.
Copart, can you rotate me up?
Yep.
Just
Got
to
the
center
right
up the
sub-branching.
branches.
But it doesn't seem
Slight
to have
tilt up.
all the main branches coming out in a
Stopped.
rotational pattern at the top like we would expect for the genus Umbellapathes.
Yeah, but they're not all coming out in a plume, though, Scott. They're coming out in
Can you
several
tilt up?
different heights
Tilt up?
along the main stem.
Yeah, go for it.
Right, that's what I'm saying. They're
Yeah.
not coming out in that
Hold
pattern
there.
Stopped.
of a ring near the top.
So I'm going to throw this out because you need to go fast on this. Is this a collection possibility for folks? Anyone like to weigh in on this? At least from my point of view, I've never seen a black coral like this. Tina, unfortunately, is not on the line. Tina Molitsova. But what do you think, Scott? Do you think this is kind of common, or is this new enough to warrant?
No, it's not common. I don't recognize it. I'm having a little bit of trouble placing it right now. It's got elements that remind me
Tilt down
of
full screen.
Slight down.
maybe Lillipathes, but
Just slight.
the color is off. Maybe it's just a
Not
really young one,
that
but
back.
I think it's worthy of a collection. It's not something.
I think we're okay to
that
collect.
can down easily.
I mean,
Tina
I'll
is
probably
on the
right over
line.
top of you, but
Excuse me. I just
there's
see her,
nothing
and
to run
she's
into,
suggesting
so.
Lillipathes. Tina, what's your opinion here, and how about from some
Yeah.
of the other How about anyone else here who wants to weigh in?
Lillipathes, which is the closest I could suggest.
Yeah, we're still waiting for a consensus. Sako Matsumoto's on the line, another
Can't be ahead of
octocoral
time. If you
specialist, Les
turn,
Watling. Now we
might
got
be
a
able
whole
to
bunch
open
of
up your
people in
box without
the
lifting
chat
it.
room.
Sticking out loud.
We have. What do
I
you
gotcha.
think, guys? Is this a good candidate? Let's get some more
Or if you're
opinions
stable like
going.
this, I'll just grab it.
I think I'd have to I'm going to move closer to it if I was going to grab it.
Yeah. copy.
Maybe pivot, turn left, lateral right.
Can we look at the branch to the right?
Absolutely. Video coming right.
It's the one that's closest to us.
This one.
That one, yes. Max zoom.
Copy.
Well, Scott, we're not getting a whole lot of other responses here, and we have to make a decision pretty fast. So I think it's worthy if the pilots can.
One out full
They're in
wide.
a position to grab this thing. I think.
Yeah,
this
if you
could
want
be
it, we
good.
can certainly give it a go. I'll get set up for it.
Yeah, we're waiting. We're waiting for Tina Molitsova, who just got on the event log. She's at the Shirshov Institute in Moscow, and she's a specialist in these black corals, and she's looking at it very closely right now, which I think is why there's why we're waiting to hear from her. She's thinking about it. Les Watling thinks it should be collected. Looks very strange, and Tina says yes, perhaps if we could grab it. Certainly never seen this type of black coral, I don't think, in the Hawaii Johnston area. I can't remember ever seeing anything like it. And we don't have anything in the animal guide that looks like this. Scott had thrown out, let's see, he's actually looking in the guide right now, the Okeanos online guide. And I'm clicking on his link.
Yeah, video, you can frame it up if you'd like. Make sure I'm not moving.
Yeah, what I sent you
Yeah,
would be a
hydraulics up,
much
co-pilot.
older
hydraulics
colony
up.
than what we're looking at here, but you can
yeah.
see the coloration is different. in
Yeah,
terms
we'll
of
just have
polyps,
to-
and I don't know
yeah,
if
you have
that's
to
just
lift off.
an effect of age or feeding.
Well,
But that's the closest I can
might be able to get a little drawer open.
see
I don't
to
know
what
how
we're
much.
looking at.
Okay. I could try pivoting around.
Yeah, so
that too.
Tina
Well,
said
it's
it
up
is quite possibly new, and those are
You
the
can float into it.
magical
There's
words
not-
that we love to hear.
really anything for you to run into. Too.
So-
It's up to you.
Folks,
a
those
lot.
of you who are watching in the public, we may be looking at a brand new species of science here, so we're going to go ahead and collect it. We won't know until it's given a very thorough examination in the laboratory. But
What camera?
you may be seeing it for the first time as we
Bring
are.
me Zeus, right? Right. Yeah.
Zeus, you got it.
Okay, watch this. We're gonna go for the collection. I'm thinking just snip the top portion,
Yep, maybe about halfway down.
halfway down.
Yeah, so
Okay, I'll get close to it, and then you'll
yeah, just
Oh.
make sure we
Maybe
get
this is
enough
something
to-
tissue
Yeah.
here to,
Craft.
for sure.
because we need a bunch of these branch
All right,
points on
there
it
you go.
as well.
Yeah, I think that's well said, Chris. Some of those branching points are going to be important, but try to leave at least the lower branches so that this thing can continue to grow.
So upper branches, leave some lowers. Doing the center with mini Zeus.
Yeah, that video can come in on Mini Zeus a little bit. Yeah. Okay, so maybe.
You might, yeah, that's what I'm.
Did you guys get the
thinking.
science description about leaving the lower branches for
Yeah, so
regrowth?
we're gonna try to leave.
Yeah, I think that looks like a good height.
Well, actually, does this look like a good position to trim from here?
Yes,
I think that'd
it
give you
is.
enough. Check my grip force first.
Nicola, I just hit escape and it came back, so we're good.
5.
So we're going to call this Lillipathes,
I can tilt up real quick.
yeah,
on
Lily,
Zeus.
sort of just like, but with an I,
Port manip looks like
It's
you're
L-
not quite
one L,
far
I
enough
think.
in.
if I'm not mistaken, but I'll check on that. Wait, actually, yeah, 2 L's, you're right. Tina's saying close to Piranopathes and Lillipathes. We'll just call it Lillipathes with a question mark. And then, Pilot, do you know what box you're thinking of putting this in?
Not yet.
Okay.
Just want to make sure I get it in the coral cutter zone here.
Yeah.
This
that looks a lot
particular
better.
coral is very bendy, as you can see, and he's leaving enough, so it's
Nice.
definitely going to survive and grow back. This is great. Nice job.
Let's
And
go port inner. I'll probably take off. That way the...
Did you catch that? They're going to go port inert on that. Right.
Copy.
Sound good, co-pilot?
So
Yeah,
you're hearing
sounds
us
good.
talk a little bit here in the
I'm ready for
background. So what's happening is at the very moment of collection, then we've got a screen up in front of us, and we're putting in some information, a specimen number, where it's actually going, a tentative field ID, and then once it is secure in the box,
Let me know when you're
we
ready.
press a button, and it
We're extending
captures the
drawer now.
longitude and depth and all types of information with this collection.
All right, you're good. I'll come down on Mini Zeus.
We never do
There you
this
go.
until it's in the box, because you never know what can happen until that box closes shut, and we've got it for sure.
I was just making a comment in the event log that the genera Parantopathes and Lillipathes don't separate genetically, and she's referring to work that a previous PhD student of mine had done, Mercer Brugler, and using the genetic markers that we had available at
that's
the
good.
time, it was very difficult
Nice,
to
thanks for the
sort
bottom.
out
It's in.
the differences between these
Good job.
taxa, even though morphologically they were really quite different.
Thanks.
So we're still investigating the genetics of that.
Yeah, thank you. It is in the box, folks. We've got it. And it's, at least for the moment, it's stumping the coral biologists out there. So this is great. This is the type of animal we do like to try to find.
There you are,
So
pilot.
thank you very much, Pilot. Excellent job.
Yeah, thank you.
All right. Hydraulic secured.
Okay, we're moving on after we've got that specimen. And I think it was Roland who spotted that very hard to see wispy colony. So thank you, Roland, for your sharp eyes on that. There's a little bit of a different sponge there.
Copy that.
To the left. It's the first time we've seen this variety of glass sponge. A snap zoom would be sufficient, I think.
Okay, video, go ahead and snap zoom.
Oh, yes. Oh,
Watch your
hey, could we get a little closer on this by any chance?
Absolutely. You probably want this side of it, I think.
Yeah, this side is the right side.
Coming around, coming in.
This is really great.
Some other colonies below me, so I probably won't sit down for this one. Is this close enough? We have additional zoom here. So
just a quick additional zoom.
Yeah, video.
That's it.
I'll do this floating.
And that's, well, there's a little critter in there.
Amphipod, I'm guessing.
Yeah, so this is actually in what we call hard sponge group. I think it's in the family Phariidae, and it could be something that's called Spitosulia. This identification, if it's correct, is coming, if I'm looking at the same thing, is coming from Dr. Konstantin Tabachnik, who's also at the Shirshov Institute in Moscow with Tina Molotsova. And so thank you very much. That's as good as we can get on this. There's a little ophiurite, isn't it?
Yeah, I think so. Looks like it's hanging on the outside.
Is there anything you'd like a closer look at on this specimen, or?
No, thank you.
Okay.
That shows
Yeah,
me
the top.
what I need for
Okay.
the identification.
All right, video. Yeah, we'll come all the way up to the top, and then we'll be clear. Okay. Yeah.
Quite a name. I never even knew of that genus until Konstantin had responded looking at
Video,
some of
let's
our
snap on.
pictures.
Oh,
Moving
cucumber.
subject.
Oh, he's swimming too.
Is this Hansenopteria-ish?
I think so. It's in the family Synallactid, isn't it? Synallactidae?
Looks like it. So this is a sea cucumber. This is a different kind of sea cucumber than the ones
get
people
closer,
might
video.
be
Yep.
used to seeing in shallow
Tracking
water
focus.
habitats.
Copy.
But they're fundamentally the same type of animal, except that in the deep sea, several of these can actually take off from the bottom and swim. This one is not primarily a swimmer. It's what's called facultatively benthopelagic. That is to say, it lives on the bottom and can take off
watch
into the
your,
water
starboard
when it
toe
wants
as you
to
come
find,
forward.
presumably, newer foods to eat. You can actually see the food it's been digesting through its body wall because the body is transparent, and you
It's
can
settling.
literally look at the gut contents in the gut. And so that's what that kind of gold tube is.
Still
So
pulling.
after swimming, it's
Yeah.
now sort of settling out. And
I don't want to back into anything.
More
So.
tilt.
That's about as far as I could swim too, so I'd be pretty
There you
tired.
well, I think it needs to
We're
sort
gonna let
of
it go, video.
Swimming crinoid bottom screen.
leave its. Wow, there's all sorts of
Yeah.
swimming echinoderms today.
Well,
So
back
here's
looks
a
safe.
swimming feather
Copy.
star, also called a crinoid.
Sit
These
down.
are also
Rotate slightly forward to keep you in view.
sort of. They're not primarily swimmers,
Thank
at
you,
least as far
co-pilot.
as we know.
Lasers are clear.
But they will, when
Lasers
provoked,
are
start
off.
undulating their arms and kind of achieve some
C-pin
kind of
also.
lift as they swim up into the water column. This one might be doing so in reaction to our presence.
Considering
It's not
how
hard.
to
It's
zoom
not
more.
clear. But it's thought that the swimming behavior might have been evolved as an adaptation
It's coming
to
at you.
similarly slow
Yeah,
predators,
get closer
such
to.
as sea urchins. And this doesn't help a feather star if it's trying to escape from a fish, but it does
Still
help
coming
if
at
it's
you.
trying to
Yeah.
escape from something that is sort of working in its identical time frame. But
Okay, we'll let it go, video.
only some feather stars have the flexibility in their
All right.
arms to actually do this. So that's neat. Today we saw there's at least two
See the sea pin?
groups of echinoderms, spiny skinned animals
All right.
related to sea stars,
Good snap.
and within moments of one another, we've seen them swimming through the water. So pretty neat. Oh, and here's is this a rock pen?
Yeah. I actually already logged it, the rock pen, as we were getting the sea cucumber. So maybe Anthoptylum, it's really not clear to me, but these are very large polyps coming off of a main branch.
Hi, video.
And there's a black
Yeah,
coral.
these are really difficult to identify, even to genus, without having the specimen
Oh,
under a microscope. But Anthoptylum or
Getting right between the skids for you, video.
Cophobelemnon, Califolemnon, those are all possibilities for these rock pens.
Come back in
It
video.
looks like maybe, 3 hydrozoan colonies sticking off the rock face just behind that rock pen.
Yeah, there are, and there's a black coral next to the Holothroidia as well. Just lots of animals here.
Yeah, I thought that might have been
Sorry
another
about that video.
heteropathes.
Here we go.
Nice view of the kind of suction base of the rock pen there.
And there's a beautiful view of the Holothroidian right now showing, those are pseudopodia, is that what you call them? Or what do you call
Those are the
things?
tube feet.
Tube feet,
Yeah,
sorry.
sort of looks like they're around the fringe, and it looks like we're getting a classic view of where the sea cucumber deposits its sediment when it's done with it.
But what do you call these things on the top?
I think those are actually, I don't know what they're called, but they're modified tube feet, if I remember correctly.
Okay.
Okay, we're clear.
All right. Just pull wide.
Yeah. Thank you very much.
Looks like there's a worm or something on that black coral as well in the background. I wonder if it's another aplecopteran or if it's just another scale worm. Scale worms are more commonly observed on the black coral.
Which black coral?
oh,
Oh,
the one
those
we're
are
looking at, yeah.
Very small white. We can come back in. We can.
Yeah, we can
Get it
do
real
a
quick.
quick snap zoom on it.
So you got video?
Yep. Know what that is? Maybe a snail even?
Is
Not
that
sure.
full zoom video? Yeah, give me full zoom and I'll try that. Oh, here we go. All right. Stand by one as I center it up. Copilot, rotate up about for me.
Rotating up. Stopped. Oop. Rotate down.
Is it a worm maybe?
Stopped.
It's a
Yeah,
slippery.
it's a worm, I think.
Slide up.
Isn't it?
Slide.
Yeah, polychaete. Sure.
Yeah, so it looks like a little tiny polychaete worm living on this
Worked great.
black coral.
Okay, video can come wide when you're clear.
Saw some similar
It's a
to
slippery
that
rock
on,
that I was putting my
was
foot
it the
on.
Hemicorallium or was it the
Thanks for
Chrysogorgia that we
Smooth.
collected the other
Not
day?
ideal.
Yeah, it's
I
Chrysogorgia.
rotated due
Chrysogorgia.
south,
Yeah.
about 25 degrees to port.
Lehman Charlie.
Yeah,
Center
there's a
back up.
family of
Yes, if
polychaetes,
you
the
center with me, I'll just kinda
Polynoidae,
rotate.
that are the more commonly called the scale
As you
worm,
come center.
that have a lot of species that are associated with many different kinds of corals and with many other kinds of invertebrates as well. They have some specializations that allow them to live very closely associated with other animals.
We have some big bamboos in the background already, so the density of animals is definitely picking up here.
All right, we're back on track here. So possible bolosoma.
Yes, I think. Yeah, maybe. Not so sure. We could do at least a snap zoom on this guy too.
Yeah,
oh
but-
yeah, I think you're right. Way to go.
Video, we can go snap on this, just to get an idea.
Yeah, it is. It's kind of a little bit weirder texture, kind of more bumpy.
Coming in for landing.
That's a weird bolosoma. It's got some very large osculum on the surface of it.
Here's another one of these little tiny crinoids, these feather stars, that's living on this one as well.
But yeah, I completely agree. Bolosomini in that subfamily, the stalked euplectellids, and bolosoma is a pretty good guess to the species.
So for people who are just tuning in, we're currently looking
Yeah,
at
video.
a glass sponge.
Video
So this
clear
is an
below.
animal that's actually made out of glass. And its skeletal structure is biogenically developed by the animal, and it
Lasers on?
has a stock
Lasers
that looks
are
very similar
on.
to fiber optic cable, and the skeleton
I'll get him right next to
of
the
the
base.
body is like
There you go.
sort of a lacy, glassy
video's
structure
clear.
unto
All
itself.
right.
Remarkable
Thank you,
deep sea
video. I'm
animal.
going to keep moving. See a couple large colonies up here on my left.
Yeah, do you want me to rotate?
Yeah, I think they're going to want to see these.
Agree, pilot. Thank you.
Yeah, definitely.
Copy, rotating.
Think I got enough tether to get there?
You might. I can come down a little bit as you go forward.
All right.
Let me know, co-pilot, if you're thinking about another ship move.
Okay. We'll get these. We'll see if I have enough tether. Pretty large colonies, huh?
Do we have lasers as to how big they are?
They are, yeah. I'd say the base of the center one is about 10. Here's the lasers. I'll bring them over to
Wow.
the white zone there. Gives you an idea. Yeah.
Obviously, a good place to live up here on the top of this feature.
It's good to
And
be
one
the
of the
king.
things we're often wondering is, when you see these really, really large colonies, what are all the factors that went into allowing them to survive here? So the currents must be strong enough to deliver them food. They must have been good enough for the larvae
that's
to get
about,
here in the first
yeah.
place
All the tether I give
and
you.
not get
Copy
buried.
that co-pilot video. You
And
can
I wonder, are these
come in, maybe we'll
three
get the
related to each
black
other, or are they all
background of this
independent
contrast.
colonizations?
Can't,
And I'm looking at these two large bamboo corals in the foreground. I'm also looking
You
at
got
the
a small
background to
movement?
the right, and I'm wondering if that one in the right is a different species
Yeah,
from
okay.
this. It has much
Can
more
we do 10
angular
meters at
branches in
200?
the background.
Copy that.
Move me a little closer.
Okay.
This one is also. Oh, this is branching from the
Great,
internodes,
now.
I see. So they are
Go
definitely
ahead,
different
Nav.
species.
Let's
Oh,
get another
interesting.
move in.
Okay.
It's going to be
Background is what we
10
looked at before,
meters,
that branch
bearing
from the node.
200, speed 0.2.
So which one is this, Scott?
Well,
Seven
we
up.
would put it in the
Can you repeat
genus
that?
Keratoisis for the time being,
Range,
but those
10
names
meters,
are going
bearing
to be
200,
modified because we now know that
speed
there are actually
0.2.
several
That
different
is
genera
range 10
that are
meters,
subsumed
meters
within
bearing
that one
200,
name.
speed decimal 2.
That
And
is correct.
as we're kind of looking over here, get a clue from the sclerites themselves to see if I can give you any further clues. So everybody can look with me. I'm not seeing the really sharp needles protruding between the tentacles, and I am seeing what look like
Is that full zoom video?
shorter rods in the
Yes,
body
yes,
of the
copy.
polyps, and so those are important clues.
RV nav ridge, move of 10 meters bearing 200, speed decimal
Speed
2
to top.
initiated.
Thank you, Bridge.
All right. Co-pilot, can you grab my rotator?
Absolutely.
Let's just come up slow all the way to the top.
Coming up slow.
Looking for little spikes.
It's possible this is in what we would call the B clade.
Okay, the D-clade.
But B is in boy, B is in Bob.
B.
Bamboo.
Anything
There
there?
we
You
go.
want to keep coming?
Yeah, let's come up so we get to the tippy top.
But
Top
yeah, tough for me
eye.
to say right now.
So, what do
Alright.
these eat exactly? I mean, it looks like they've got mouths
Oh,
everywhere.
That's good.
Is it just organics, or do they actually feed on
Okay.
things?
Video's happy. I'm happy.
Yeah, so every one of these polyps has its own mouth and its own tentacles that are presumably used to grasp things. And it's something that, we've asked ourselves a lot. What do they feed on? Because we never get to observe a predation event. We sure would like to. And there have not been that many studies that have directly tried to assess what they're feeding on, at least in the deep sea. And so studies have to rely on either comparison to shallow water, where
You
they
want
are
to zoom out a little
known
bit,
to
see if we
feed
see
on,
any associates on this.
and when I say they,
Thank you.
I mean
Oh,
Only associate I've seen was a very small little crawler
on
on it.
very small zooplankton.
Yeah, I think I saw him as we were
that
coming
swim
up,
by and they
or
eat grass.
rotating up.
And
But nothing.
Les Watling, one of his
Something on that
students, Les
left,
Watling
Yeah,
at
I think I'm see what you're saying.
University
Slightly
of
red in
Hawaii,
color at
one
the
of
tip
his
of a
students
branch.
when he was at the University of
Setting
Maine
it
was
up
doing
now.
some studies on reproduction in these deep sea
See if I
octocorals.
don't lose
So
it.
she collected a lot of polyps and then embedded them and did some thin sections. And we saw
yeah.
in some of those thin sections
Rotate down
small
about.
crustaceans in
Yeah.
the guts.
Rotating.
So we
Small
know
red
they
in color.
can feed, at least some of them can feed on
down.
zooplankton. But there's a question about,
Yeah.
maybe they're just feeding on organics,
Yeah.
Stopped.
Is that
bacteria,
situation?
all
Looks like
It's a really good question.
Hey, Scott. We're coming up on some
scroll more
benthic
left.
ctenophores
a
here.
copy. Coming to.
Oh, wonderful.
Might be a-
So, yeah,
This is also,
if we... Sorry, go on.
go ahead, Chris. I don't know if you want to describe them or if you want me to.
Oh, no, please go ahead.
Well, so ctenophores are very commonly observed in the water column. They're sometimes called sea
Go ahead and take it to the
gooseberries.
far
They're
right edge of the screen so you can
little
see the tentacle.
jellyfish-like animals that
far as
swim
possible.
using
Oh, they're pretty
cilia,
long, aren't
rows
they?
of cilia that
Yeah.
beat
Off
kind of
screen
like
right
the
now.
rows of the oars
Yeah,
of a boat.
I'll try that.
But this group of ctenophores, called the platypennid
Okay.
ctenophores, are specialized to
okay,
actually
you
be
still wanna
benthic.
keep
That
the-
is, they are sitting on the surface of the bottom. Usually they're sitting on some kind of animal, and it's not unusual
there's-
for us to see them on corals or on sponges. I don't actually recognize the morphology of these species, but one of the things you can see, there are these little orange structures back there, and you see there's an extension,
Do you
kind of
wanna
like a little
rotate?
chimney
Yeah, I get it
coming
now.
from them, and then there's a fine line coming out of that
Slow
chimney.
down.
So those are, the chimneys are called
there's a
tentacle
stop.
sheaths.
There's that other
and
colony
all
is in the
ctenophores,
foreground right
or most
now,
ctenophores have
fortunately,
these
but-
tentacle sheaths. And then they extend the tentacles kind of like fishing line, and embedded in the tentacles are specialized cells that have structures called coloblast, and it's kind of like little packets of glue, so that
Slide up.
things that are swimming by and
Stops.
intercept those tentacles will cause those glue packets to fire, and then the tentacles can be withdrawn and they feed on them.
So
Interestingly, the mouth is
it's more like a
actually
sticky-
amplified against the colony
it's-
itself.
So it's
That's
sticky packets, not stinging cells. Scott? Still with me?
Yeah, I didn't hear the question, Chris.
Oh, I'm sorry. So they're not stinging cells like you would find in jellyfish. They're actually like sticky
Okay.
packets.
Or pull for
Yeah.
full
That
specimen
is correct.
colonies.
Right. Ctenophores do not have the
To rotate
stinging
down as it
cells
pulls
and
out.
nematocysts
Yeah.
that all the
Down,
bacteria have.
coming down.
Instead, it's a
Nope,
specialized kind of
stopped.
cell only known from the ctenophores that
Interesting.
is sticky.
Okay, so we're now pulling back on the bamboo coral. Now, did you say that there was another one that
That
you
is
thought was a
full
different
line.
species?
Copy.
Yeah, in the background on the right, it's way in the background, and all I was pointing out is if you look at the one in the background, you'll see that the branches arise at a much sharper angle. These ones, the ones that we were just looking at here, they kind of curve nicely, gentle curves.
Okay, so you're talking about these
That's
living
correct.
ones, or are you talking about one of the dead ones?
No, not a dead one. You'd have to turn more to the right.
All right. I think you saw about the one
There
of the
it is.
four in the background, the darker in color.
Note,
because
note how angular in
yeah.
the background that colony, the branches come
see
out at
that
a
one?
sharper angle.
yeah.
Okay.
Go ahead, video. I think
Pilot, do you see
think
that?
this
You
is
don't
the
have to go
one.
over to it. I think it's
Yeah.
the thing I called Isodella earlier.
Oh, okay, so you've identified it already, Isodella? Okay. So yeah,
Correct.
this is fine, I think.
Okay.
Yeah.
Great.
We don't need to go over there.
Excellent.
All right.
Okay, video, and you're clear.
Yeah.
I just wanted to establish and chat. Okay.
Lovely. Thank you.
All right.
Yeah, I think
All right,
we're.
you have
your
Scott,
consider
does that suit you?
Yes.
you should,
Yeah, that's good for me.
Okay,
I was just trying to
short
raise
of that
everybody's
10-minute
attention
movie, so
to
if you wanna
some of
put
these
another one in.
characteristics, like how the branches
yes.
arise, gives us a
It's
clue
not,
to what the
Pilot, you want us to put another move in?
identification is.
While
Ah,
you
very good.
Yeah.
Okay.
Go ahead.
copy.
All right, pilot, we can move on.
Copy that. Let's
Thank
snap.
you.
Roland, I think as we were talking about, snap on this, see if there's interest. See if they see the possible anemone or something.
Ah, a little sea anemone.
20 meters at
Quite a big sea anemone, actually. So, for people who are watching, we have these lasers that are on screen that help us to determine the size of what we're
190.
looking at, since we're often zooming and
repeat
zooming in. And
20
the
meters,
lasers are
190.
10 centimeters apart. So, comparatively, that sea anemone we're looking at is a fairly nice dinner plate-sized animal.
Okay, video, come wide. Let's see what else we got in this
Lovely.
area. I don't have
20 meters,
Thank you.
190 coming up.
Okay.
Bridge, this is nav.
Go ahead
I
now.
think that's a similar
Let's
sponge.
get another move in. That's
Um,
gonna be range 20
the
meters.
sponge stocks, I
bearing
think we can
190.
Move on from.
Speed 0.2.
And
Range of 20 meters, bearing 190, speed decimal 2.
That is correct.
yeah, let's continue
Swimming
on course.
center screen. Fish.
Oh, we kind of
Zoom out will Roland, but I think he's gonna be gone pretty soon. Go ahead, Roland. Yeah, he's not cooperating.
ROV nav bridge, move of 20 meters, bearing 190, speed decimal 2.
typical fish,
initiated.
all uncooperative.
Copy, bridge. Thank
Yeah.
you.
You're welcome.
We'll let it go, Ron.
Yeah.
Unless it comes back in the view or something. But
Okay, co-pilot.
Second
starting
fish I've
moving,
seen
should
today.
Feel the swing probably at 21-24.
Copy that. Thank you.
okay, this is the direction of movement here. I'm lined up with you.
Yep, it's pretty good.
So if you're just tuning in, this is Chris Ma from the Smithsonian's National Museum of Natural History. This is aboard the Okeanos Explorer. This is part of the La Lima Oka Moana expedition. We're currently investigating the Johnston Atoll Unit as part of the Pacific Islands National Monument. We're currently at a depth of 2,170 meters.
Nice video.
We're looking at deep sea
Pleasure as
habitats
always.
that are predominated by sponges and especially different types of deep sea corals.
Watch, is you interested in this possible small white sponge?
Yes, let's take a look at it.
Just caught my eyes.
Thank you.
Um,
And then I'm not sure if
okay,
that,
video, we can come in and see what we got here.
that red
Can you guys
spot
see
in the background.
Thank you. Welcome to the chair, by the way.
That's interesting.
You can come in further if you got it. Do you guys need a closer look at this, or it's pretty small? Hard to see.
No, I think it's a sponge. I don't recognize it.
Okay.
But yeah, I think we can move on.
All right, video, come out.
And is that red spot, is that a shrimp? Let's take a look at that red spot in the background.
Yeah.
To the
Good
left.
snap-on shrimp.
Thank you.
As I come in, I'm getting slightly closer to it, so focus will change.
Okay. This looks like one of those long-legged shrimp that we see throughout this area. I believe Chris called it nematocarcinus. We can move on. Thank you.
Okay. All right, video. Let's see what else we got here. Have we seen this coral?
What's... Oh,
Yeah.
well, the big yellow things I think are dead sponge.
Okay.
And then this,
Yeah.
you mean this
Spirals.
the spiral? Oh, yeah, let's take a look at the spiral.
I think it's going to be...
Yeah, I was just going to point that out myself. I'm wondering if that's a chrysogorgia. Maybe an iridogorgia,
Maybe you can
perhaps?
frame it up.
Sounds
I was
good.
just about to say Iridogorgia,
All right, you can come in.
but I
Zoom
I
at
don't
will,
know.
if you will.
Well, thanks.
Take it. I got a good foothold, I think.
Okay, so what say ye, Scott?
Is this an octocoral?
Yes, it is.
I am still waiting for a close-up.
Ah, okay.
And naturally, I just ran out of computer power. Stand by.
Okay, so we're definitely looking at an octocoral. Everybody, we're looking at a subgroup of the
214.
greater sort of cnidarians, which are animals with stinging cells. These, in particular, are known for having
And that's full
8
zoom.
feeding tentacles
All right.
around the mouth. And so what we're zooming in on are polyps. This is a colonial animal, and so these are feeding structures that all contribute to the greater colony, which is part of this spiral body. And at the moment, I believe Scott's currently researching what type of animal this is amidst his technical challenges. And hopefully we'll hear back from him shortly.
ROV nav bridge move
What did
complete.
the people in the chat room...
Sorry about that, Chris.
Copy bridge.
I was working off a laptop and I forgot to plug it in, and naturally it went off right at the wrong time. So yes, indeed, this is in the family Chrysogorgiidae, which translates to the golden corals because of the nature of the skeleton. And this is in the genus
All right, Copilot, you click my rotator, maybe
which is
find the top of
characterized
it.
by having this
Got it.
nice
Seen this
swooping,
pretty good, I think.
spiraling
Maybe
main
associates.
axis with
Coming
then
out.
these individual
there's a
Oh,
ridges
feeding,
on the
almost.
axis.
Yeah.
You can see the
Yeah.
polyps are kind of
Follow
delicately
that little
arrayed
guy.
on top of
Yeah.
these side branches. One of the things I always find interesting about that Chrysogorgiidae is that the tissue that covers the skeleton is extremely
Yeah,
thin,
chase
and so the polyps
small
really
movements of it,
stand out.
maybe.
And that's true also of a
Distraction
lot of
shots.
the bamboo corals, but it's not true of some of the other sea fens that hopefully we'll get to see later in the dive. This, I think, is the first one that we've seen, possibly, on this expedition.
Yeah, I don't think I've seen... Well, I think we've seen Iridogorgia before, but I don't know if we've seen it today or even recently. It looks like it's actually caught a little crustacean. We were just asking about how they feed.
Wish I was closer. We're max zoom, huh?
Yeah, that's
That is
as
fantastic.
far
I
as
see
it
what
can
you
go.
mean.
Copy.
I did see the crustacean swimming, and I saw a couple of the tentacles curl in over it.
Yes,
I think it's what, the third polyp from the main stem on that upper branch?
correct. And I think
Wow.
it escaped one of the others, which is closed now, the one that was on the bottom. But I think that third polyp there has held
Sorry,
it fast.
rotate
And
Cirrus real quick.
we're seeing,
All right.
this might be one of the first times we've ever seen this species feed.
That is crazy. This is so funny because we were talking about never seeing it.
Yeah,
Maybe
we should just
rotator
keep
again.
on talking about the stuff we've never seen before.
Still centered, right?
Yeah, we need to continue to mention
Yeah.
other things that we haven't
He's
seen.
stuck
so that
in the
doesn't
polyp,
happen.
polyp gout carnivorous.
But is
Well,
it?
I'm going to look forward to watching that replay.
Yeah. But it has, I think, it's not drawing it in and devouring it. I wonder if it's just needs to settle if the stinging needs to be more effective before it can actually digest.
I could try to get a
Well,
different
an
angle.
alternative view there is that there's a crustacean swimming by that regularly alights on the coral and maybe just disturbs the polyp,
Oh,
and so the
close.
polyp retracts somewhat.
Yeah, you're right.
Perhaps it's not trying to feed on it, it's
struggle.
just kind of being
Maybe it's got
disturbed.
like 1 foot,
and
just
so
trying
we
to
have
get
to
away.
see that a couple of times
Oh,
to see what actually
yeah.
happens.
Looks
to
like
the
it's gonna get
plankton.
up.
What is this thing in the background? Also, or in the foreground, I think there's something on this polyp that.
Yeah, the pink thing?
Yeah, it's sort of just
Oh, there.
extended from the. Yeah. What is that?
I am not sure. It's not in focus to me.
We're at max zoom. I could reset and
okay.
get closer, but
I think it's good to. Chris, do you see this?
It sort of looks like
Yeah,
a
I
leech.
saw that, and
yeah.
it's another
Do
one
it
of
out.
these who knows,
Rotate up.
probably who knows two for today.
Good. Thanks.
Yeah, you're welcome. I'm glad to help.
Polyps on that branch all closed too.
Yeah.
Huh.
That's
Man.
unusual. I see now. Yeah, I can't tell if that's a little hydrozoan or.
It looks like it's emerging from the mouth. Huh.
Watch these. Would you like me to try to get a closer look at this? I could get closer to the subject if you'd like.
Sure.
Yes,
yeah.
please. Thank
Have
you.
you come wide as I.
This is like a
Alright.
very happening Iridogorgia today.
We're full wide.
All right.
Frankly, it
Rotator.
looked like it was sticking its tongue out at us, but I'm not really sure. Polyps don't have tongues, do they?
I don't think so.
You
I think
don't
it's
think they do.
pretty much a straight shot to the stomach in there.
I think you're right.
Okay, video, come back in.
All right.
It's about as close as we're gonna get here.
Do you want to take a rotator?
Yeah, go ahead, take rotator.
So for people who are tuning in, this is a really interesting microcosm of coral biology
Rotating
life
down.
here. We're kind of trying to get some really tight zooms on these very
Stop.
bizarre feeding and other things that we're watching.
Let me know if you want.
Some of
Yeah,
them are,
rotate up to that
Up.
unknown subject.
yeah, that sort of
lost
like
it.
almost like a
Right.
Here he is.
Oh, I see it. Okay.
I'm just wondering
Alright,
if
is that
there's
max zoom?
Yep. that's
Okay,
max.
that's about as close as we can
hydrozoan
get. Watch
that's
it.
growing just behind the
no,
polyp.
those
and
are-
it looks
there
like
we
it's
are.
coming
There we
I'd
are.
say that is plenty. Wow, I don't even.
coming out of the mouth, huh?
What was that?
You thought it was coming out of the mouth?
Yeah, it's this worm-like structure, and the base of the stalk is coming directly, I think, or close to, out of the oral opening. There's a string that's coming out of the tip of that sort of pod-like structure.
Rotate up a bump, maybe.
Huh.
And so,
Yeah.
it's not
Actually, we should
unusual
go pretty
for
far.
hydrozoan polyps
That's
in
good.
general
That's
to
perfect.
have
Down
tentacle-like
now.
structures that arise from the
Might-
interior of the base of the
the colony,
polyp
I
and
think,
are
is gonna
admitted through the mouth, and they're
float
used
around a
in
little bit.
defensive
Copy.
In my thruster
struggles,
wash.
if you will. But I've never seen one associated with an octocoral,
Huh.
and it wouldn't be bulbous at the tip. So, I don't know if this is something that is emerging from that coral polyp, or the coral polyp is trying to feed on something and kind of pull it in.
Yeah. Oh, I think that's a very good question. As Les Watling has mentioned, who is eating who, and what is this thing? This is the fun of deep sea biology. Is a lot of these things are completely unfamiliar to us, and understanding what they are is just a big mystery sometimes.
Okay, watch this. We clear on this subject?
Yeah.
Yeah, we're clear.
Okay, video come wide. We'll
All right.
keep moving here.
All right. If you're
We're full wide.
back,
Interesting.
but I have you at about. Let's see. That's 7.5 right there.
Looks like it's 11:30.
Yeah.
Oh, Chris. Okay. We're going to do a quick pilot change. Watch leads.
Roger that. And while the pilots are switching out here, so the ones who've just been on the stick can get a bite to eat. Just wanted to point out that we got 21 people in the chat room right now, and I'm going to read off some of these names here. We've got Jessica Robinson, Amy Bowman, Asako Matsumoto, Chris and myself, of course. There's somebody at the ISC Watch too. I wonder if that's Nick Polenko. There's Jamie Aubrey, and Jamie Aubrey is in Scott's lab down in U of L at Lafayette. John Smith, the geologist with the University of Hawaii School of Ocean and Earth Science Technology, is with us. Good to see you, John. And he was also part of the Falcor mapping group that mapped the Seamount that we're on today. Jonathan Tree, as I already mentioned, was on that cruise. Kelly Elliott, our expedition manager. Kevin Cocott, Les Watling, Luke McMartin, and he's calling in from Hui, and he's very interested in octocorals and their associates. Mashkor Malik, who's our mapping head, he's also monitoring what's going on. Nicola Rodriguez, who's to my left here, who's just been fantastic with annotations and helping with specimen processing and all types of things. You've got Nolan Barrett, Roland Bryan, of course, Scott France, who you're hearing his voice all day, who proposed this dive site, Tara Luke, Tina Molitsova, and William Mowat. So we've got a nice group of scientists who are helping us out, scientists and coordinators and everything helping us out. And look, we are picking up the number of animals that we're seeing. A lot of these are bamboos, I think.
Yeah, I agree, Chris. And earlier, when we just a moment ago, when we were stopped before we saw the Iridogorgia, I saw some more of the long dead skeletons. And one thing that's instructive is to look at the relative thickness at the base of those dead skeletons and compare it to, for example, that really large one that we're just passing, so we can get a sense of how big was the colony when it died. And what I'm seeing is they weren't particularly big. They look like they're relatively small, those broken bits.
Okay. And pilot, there's a couple sponges. I think, even though they look a little different, I think they're the same thing. One of them's in the behind center, and the other one's over to the right. But if one of those was in your direction,
sure.
Yeah, this is a different sponge than we've seen all day. And there's actually a bamboo. On your way over, I wonder the lower right. I think Scott would be interested in this, unless you guys saw it while I was at lunch. See this fan-shaped orange thing right at the bottom of the screen?
Yeah.
Yeah, this thing I don't think at least I wasn't present when one of these was documented.
Okay, let's take a look.
Yeah, and then you could even afterward just zoom over to the sponge to the left, and that would work.
Sure. Get a dark background, stand by for Zoom video.
Sounds good.
Yeah, this is a great one of the Candelabra bamboo corals. And we see this morphology all over the world, and the ones that we have been able to collect into the morphology and genetics on, they're all the same. And this is a new genus that
let's go ahead.
I guess Les is waiting on me, so I need to get cracking. But we have a name already for this. We're all ready to publish it, and it will be a new genus.
That's full zoom.
This is one that we had collected a representative on one of the... hold on before I go any further. It looks like an interesting... Is that a zoanthid, Matt, on the left?
Yeah, I'll take a...
You know those zoanthids that we were referring... Or is that a sponge?
It's going to be either an anemone or a zoanthid, I think, Scott. I don't think it's a sponge.
Yeah, it's very lumpy. It doesn't really look like an anemone to me, but I wonder if it's something else, like egg mass that has been settled there, or if it's just there's parasitism going on, and that's actually the coral tissue that's taken on a really aberrant shape. That's very interesting.
Yeah, that's what I was wondering too.
That's something under there.
That's bizarre.
Yeah. I think that It almost looks to me like it was a basket star wrapped around the coral that has been overgrown by the coral. Weird.
I see what you're talking about. The spiral on the top could have been possible arms or something.
Yeah.
No
I
idea.
suspect that what's gone on there is something was on the branch and it's been completely overgrown by the coral tissue now. And whether that thing underneath it is alive or not is a mystery to me. I was just briefly going to say that we had collected related bamboo to this in the North Atlantic, and it was the first octocoral to have its mitochondrial genome completely sequenced.
That's great.
So just a bit of trivia.
So we are logging this as unknown. I'll refrain from calling it who knows 3. All right, we're good. We're good for science.
Okay, copy that. Let's come back in.
Oh yeah, okay. Sorry.
Just wanted to get a little bit of a different angle.
That's max.
I'm not sure it's gonna. Not sure it's gonna help.
Yeah.
This is weird.
Okay. Well, whenever you're clear video, we can do a full specimen.
It looks like it could be the tissue,
clear.
Scott. It's got a little bit of that kind of yellow-brown tint to it, like the rest of the tissue. So overgrowing something. So that sponge was to the left.
Correct.
And there he is.
There.
And
Or full wide.
Copy.
I believe this is the.
Chris, as we're zooming, you're probably ahead of me, but I was just going to say all those what look like lines or grooves that actually look like a fingerprint on the tissue. I suspect those were the mesenteries of the octocoral. And mesenteries are these kind of like little curtains that subdivide the inside of the polyp. Very interesting.
Due east 090.
Due east.
There's light wind
there's
over the bow.
probably an
no
isodella to the right there. I'm taking a
Alright.
guess as to what that is.
Copy that,
And
thanks.
this
So that's a partial.
is, I think, a Lefroyella, but I'm not 100% certain. I'm not seeing the anemones that you usually see on that type of glass sponge.
That's full.
But then Konstantin Tabachnik at the Shirshov Institute, I sent him some pictures, and he said the anemones were not necessary to go to that genus. It's more ridging and the actual structure that's really important. But I do see anemones, and they're on the other side. If you look at the edge, you can see the tentacles bobbing right on the edge on the right-hand side. You can't see the anemones themselves, but you see their tentacles. So this is definitely Lefroyella, I think, pretty comfortable.
Do you want to go to the other side, Botch Lead, or?
Well, it might be a treat for some of the public that have never seen this sponge.
You were looking for those anemones. It says how close a relationship there is between these anemones and
Come
this
on when you're
sponge.
finished video.
Clearly, this is something that has occurred over evolutionary time, and
More full wide.
we see it again and again. It's not just a
Got a
random
pretty big
placement
branching
of those anemones on this
something
sponge.
up and off to your port.
Yeah, so up
Copy that.
until
See
recently,
the
I
view?
thought that every member of this genus had to have those anemones, or you couldn't go to Lefroyella, as I said. And so, but we've also seen some sponges that didn't look like they're in this genus, but had
So
anemones
our
too.
ship move will be where video? Sorry, Nav.
about
So there's some
225 is
pretty big corals in the
should be
background,
upslope
too.
and to the
See, there's
waypoint.
this is a great
Copy.
site. But yeah, we'll just do a quick look so people can see these anemones that we're talking about a little bit better.
Okay, let's start to come in video. We're just going to do a snap zoom. I'm not going to sit down.
Alright.
And you have to go in tight to spot. That's a weird kind of morphology for the colony.
Okay.
But
Just hold there, video.
Okay.
and you'll see in a moment what we're talking about when they get a little closer. There's one weird Lefroyella. The other way the stalk is kind of almost leafy, but you can definitely see the anemones down on the stalk in particular. And you can see them up on the face, or you will in a few minutes or a few seconds.
Okay, let's come on in.
All right.
And there they are. You can see their tentacles all. It's easiest to see them right on the edge. They're kind of the little discolored balls in amongst the the tissue there, but you can definitely see the tentacles of the ones right on the edge. So yeah, just panning around, look at that. The whole sponge is covered in these things.
Okay, let me know when you're all set, video.
All right.
Yeah,
I
thank
think
you very
video's
much
clear.
for that. I really appreciate it.
All right, video's going to switch out.
Copy that. Thanks, Annie.
So,
So we're
based
gonna
on
shift
Sirius
it.
sonar, it looks like 200 is upslope rather than.
Chris, I should have said
225.
earlier when we were looking at it and you were making a notation in the event log, but that
we could
bamboo
do
coral
that
that has
or
that
somewhere in
Candelabra-like pattern is a new genus, but it's what we've been referring to as clade I4.
doesn't matter to me.
Looks like they've
So
been
if you want
moving
to
around
stay consistent
200.
with
Maybe
the names that are in the animal guide,
Sounds
clade
good.
I4.
It's 30 meters.
Great,
Okay.
thanks, Scott. And Nicola's going to update that. I just went
Ready
to
for
new
that now?
genus. I didn't have the animal guide open right at that time.
Yeah,
but
I think
thank you
so.
very much.
Let's see what they want to do
So,
here.
are these some different
Previous co-pilots said they've been
bamboos, Scott? I'm not
stopping
sure
a lot,
we've
so
seen
maybe 30 is
this
too much.
particular one.
Yeah, I think these are the ones we were imaging a little while ago. You may not have been there, but we did see
I
two
could
very
probably
large
do 20
bamboos.
right now, just based on
three
how
of them
stretched
actually
out you
close
are.
together.
Copy that.
Possibly the D clade or the B clade.
Bridge, ROV
There's
nav.
another Iridogorgia in the background, spiraling away from us.
Okay, thank you very much.
This is the bridge. Go ahead.
You know, I don't see
like
any
the ship move,
Chris's
please?
beasts on any of these so far. Oftentimes you
20
find sea
meters,
stars predating
bearing
on these type of
210,
corals, but we haven't
speed
seen that
0.2 knots.
so far today, or even on this cruise, except for
That's
that
20
shallow
meters,
dive we did.
bearing 210, speed
Yeah, it's really
0
interesting you
decimal
say that, Chris, because
Good
I
call.
remember in 2015, Les Watling and I saying we were amazed at how many of those predaceous sea stars we were seeing on the corals, far more than we saw in the Atlantic. So I'm a little bit surprised that so far we haven't seen any.
There's definitely some occurrence by bathymetric distribution too, because Circeaster tends to be more lower bathyal, not so much these deeper depths. We tend to only find one of two things here,
ROV nav,
Eoplasoma
move
and
initiated.
Hyposceria, and there could be factors that limit their distribution as well. So, but I have faith we will see predation today. I'm sure.
220.
Yeah,
210.
210.
Chris,
Copy.
when we were down here in 2015, you know, we were typically down at these depths, and I'm pretty sure we
20
saw them
meters, so
feeding
that's
here,
only
but
enough
maybe
to
I'm
catch up
mistaking
with
with what was going on in
Yeah, let's get
Papahanamo,
serious
Bulgaria.
over there.
I'll just note the bamboo corals we passed there, they're really quite large. The base of those colonies, the thickness of the branch tells us that that has been growing for a very long period of time. As they grow, the soft tissue that's covering the skeleton deposits layers of calcium carbonate. So when you cut them, it almost looks like a tree with a series of rings that are
in on this
indicative
video?
of aging.
Oh,
Although
there.
those rings aren't necessarily annular rings like they are in trees. It don't necessarily mean that every
coming.
year you get a ring.
Thank you, Scott. So
Okay.
you or Chris Ma or somebody's going to have to ID these crustaceans. I'm not very good at this at all. These
This is an amphipod.
amphipod. Okay.
It must be a
Think
female
about this guy
carrying
again.
eggs. You see how orange it is on the lower side at the front end.
Yeah, thank you.
And all of the amphipods are paracarids. They're brooders. They have these little chambers underneath the thorax where they will release the eggs after they're fertilized, and they will just hang on to them until the eggs hatch. And so then all the juveniles swim away. So they do have a form
Oh,
of parental care.
looking for a rock.
That's what you're
Scott,
looking
is
at
that
there.
midwater or is that
Okay.
sort of a bottom swimming
Yeah,
thing?
we are.
Okay.
Yeah, sorry. I thought you said we are.
That would have been a benthic one related to the benthos, so it was just
Keep my
moving up
eyes
off
out.
the bottom, but
Yeah.
that was not midwater form, not like one of the high periods.
Interesting. Thank you.
Yeah. Pilot, watch lead.
Go ahead.
Yeah. So I'm looking at the CTD screen here, and it only goes to about 1,500 meters unless I don't know a scroll function, and it's got a lot of noise on it. Is there some issue with it? I was just curious.
I can work on that for you.
Okay, thank you.
Nav will get you straightened out.
Ooh, yeah, there's quite a bit of noise on oxygen on D2. I'll work on that for you.
Yeah, thank you, Dave. You guys can introduce yourselves since you're a new group when you have a chance.